Good afternoon, everyone, and welcome back to our webinar series, the ISU UMN Research and Winemaking webinar series. We are actually the 31st webinar today. So it's a little bit too late to celebrate the 30th webinar, but it's still good to know. So thanks very much for being with us today. We hope you will enjoy this webinar and we develop knowledge and learn skills to be used in your vineyard and winery. I'm Dr. Aude Waterloo, an assistant professor and extension specialist of enology at Iowa State University, and I'm co-hosting this webinar with my friend and colleague Drew Horton, who is a research specialist of enology at the Universities of Minnesota Grapes Breeding and Enology Project. Before starting the webinar, I would like to provide you some instructions. So the webinar is currently being recorded. The recordings of the past webinars is available on YouTube following a link that I'm going to share with you later. Also, if you have any questions, please use the chat box or you can try to unmute yourself to ask your questions. We will answer your questions at the end of the webinar. Also, you're going to receive a link for a survey to complete at the end of the webinar. So please just take 2 to 5 minutes max to complete that survey. So today we will have two presentations. Lora GOULEVANT from Bucher Vaslin in California is going to share her knowledge and winemaking practices and techniques to protect and stabilize color and tannin in red wines. Then Dr. Catherine DADMUN is going to explain the science and her research projects on anthocyanins and color management in red wines made from both Vitis vinifera and interspecific grape cultivars. After these presentations, we will discuss and answer any questions you have from the audience with Anne Zwink, who is a winemaker from Soldier Creek Winery in Iowa, Mark Germany from Fireside Winery in Iowa, and Bryan Forbes from Gusmer Enterprise. But let's start with the presentation of Lora GOULEVANT.
Born and raised in the Bordeaux area, Lora has a deep-rooted passion for wine that has shaped her career. She's pursued studies in winemaking, viticulture, and wine business in the Loire Valley in France at the École Supérieure d'Agriculture d'Angers, and also abroad in South Africa, Portugal, Spain, and more, blending her academic excellence with a love for travel and exploration. With nearly a decade of experience in the wine industry, Lora's worked with renowned wineries, gaining valuable expertise. Before her current role, she was based in France at La Motte Abier, where she developed several innovative winemaking products, including alternatives to sulfur dioxide, vegan fining agents, and other cutting-edge solutions. Now product manager for enology at Bucher Vaslin, North America, Lora focuses on research, technical development, and implementing winemaking solutions. Based in Napa and Sonoma, California, she supports winemakers by offering tailored technical advice and products. Outside of her professional life, Lora enjoys making wine, surfing, and hiking in her free time. Lora, the floor is yours.
Great. Thank you very much. Thank you very much for this presentation. So I'm going to share my screen. Can you see it?
Perfect.
Okay. And all right. So let's start. Thank you very much for inviting me in this webinar. That's a very interesting subject, a very broad subject. So I will not cover everything, obviously, because it's a very broad subject. So I'm gonna try to be as much technical as I can be. I will try my best to give practical advices to protect, extract, and preserve phenolic compounds and color from grape to glass. Why color matters? First, because it reflects the wine quality, the structure, and the age potential for consumers. and it appears to be more. It appears to be a preference for the consumers to have a wine with a brighter and more intense color, especially in red wines. So when we are harvesting, when we have some grapes, we have to take into consideration several factors which create the initial potential. The initial potential is the phenolic compounds, meaning tannins and anthocyanins. They are impacted, of course, by the grape variety, the clone we're talking about, but also the maturity. So anthocyanins concentration peaks at an optimum ripeness. So harvesting underripe or overripe grapes can negatively impact the color. So having an underripe grape you will get less anthocyanins. Also, you can get more higher acidity, weaker extraction, but also overripe grapes can degrade the anthocyanins, having a softer tannin structure, and also we can lose some vibrant hues. Also, the climate, the terroir, and the grape health can impact the initial potentials. So that's the potential we have at the beginning when we are harvesting grapes. And then the idea is to extract this potential, make sure we can extract as much color as we can, protect it, but also stabilize it over the time until the wine is consumed. So first of all, let me tell you a little bit more about phenolic compounds. So in winemaking, we can observe two main families of phenolic compounds. We have the hydrolysable, which are from gallic tannins, which are gallic tannins from gallnut and taranut, or ellagic tannins. So they both are extracted from other plants, and they have specific actions. So usually the gallic tannins are used as sacrificial tannins or used on grapes because they are very antioxidant and antioxidatic, meaning that they are protecting the grapes against enzymatic oxidation, but also they are very reactive with proteins, helping to avoid botrytis and enhancing the clarification. Same with gallic tannins. So the tannins from oak alternatives or barrels are used in winemaking because they have a huge redox potential, meaning that they can prevent the wines from oxidation, but also reduction. It can fix reductions because they react with the sulfur molecules. They also help to remove some off aromas in the wine. Usually it's more during the aging. And then second family, we have the non-hydrolysables, which are the non-flavonoids and the flavonoids. The family we will talk today about will be the flavonoids. So they are present in the skin, the seeds, and the stems. So the flavonols are copigments. The anthocyanins, they are responsible for wine color. They are free, they are water soluble. That means that they are unstable. So they need to be stabilized by copigmentation or condensation. We'll see that a little bit after. And then we have the condensed tannins, which is also a pretty broad family. We have a lot of different tannins in this family, but that's the family of tannins we can find in wine. The condensed tannins are the tannins we will use that have the best action on color stabilization and polymerization, meaning that they will have an impact on the mouthfeel, reducing the astringency feeling. They also are very good to protect against oxidation and to react with proteins. This is what we will see. So first, when making red wine, the idea is to first extract as much color and tannins as we can. So we need the compounds to be easy to extract. We need to promote skin compounds extraction and limit the seed compound extraction, because usually when we extract the seeds tannins, this is when the wines becomes a little bit too harsh. So the idea is not to extract everything, but to just extract the compounds that will be useful for us in the winemaking process to stabilize the color, build some structure and mouthfeel. So there are a lot of practices to extract skin compounds. So there are a lot. So I'm just going to go through this quickly. So crushing grapes will improve the extraction of phenolic compounds from the skin and seeds, but we have to be careful with crushing because it can also promote oxidation, which can have a negatively impact on the wine quality. You can also use a cold soak method to let the anthocyanins and tannins from the skin be macerated and be extracted. It really depends on the grape variety, and you have also to be very careful on oxidation because the anthocyanins can oxidize if there is less tannin extraction. You can do saignée. So basically, saignée is juice bleeding. So you remove a portion of the juice from the tank. So it will increase the ratio skin to juice. It's a very good method. It's basically concentrating the phenolic compounds, so resulting in more extraction intensity. Then you can use SO2 because SO2 does act as a solvent, so it will extract compounds, but you really need to be cautious with that because it does have a lot of side effects. Sulfites does bleach the color, so you extract some compounds, but maybe you can extract too much because you can also extract off aromas from the skin, and it can have an inhibitory action on alcoholic or malolactic fermentations, increasing the risk of reduction or sluggish fermentation. And of course, it also does bleach anthocyanins, so it will actually reduce the overall color intensity. Then you can use extraction enzymes. So in extraction enzymes will actually help to select the different compounds to extract, such as anthocyanins, tannins, or polysaccharides without the side effects. So it can be a very good tool if you have very thick skin, grape varieties, or very difficult to extract grape varieties. And also enzyme will extract not only tannins and anthocyanins, but polysaccharides that are responsible for color stability, mouthfeel, and filtration. So that can be also a good option. Then you have the thermonic vinification or the pressure treatments. They are also very efficient, but you need to have the material, the equipment. Cap managements. So regarding the type of cap managements, especially délestage, so rack and return, pump overs, or punch downs, it will really depend that will really impact the level of extraction. Temperature really is really important in this situation, where a higher temperature will help to extract more. So there are definitely a lot of physical actions to extract the phenolic compounds. So as I said, fermentation temperature will also have a good impact on extracting the phenolic compounds and the pigments. So higher temperature extracts more phenolics and they promote more polymeric pigments. So it will help to stabilize the color. But you need to be careful on too high temperature because it can over extract the tannins and result in harsh or astringent wine. So, you know, it's always a balance to find. And then also another technique is extended macerations. So extended maceration after fermentation will increase the tannins extraction and the color intensity, but you also need to be very cautious about it because you can extract more seeds, tannins, or the unripe ones. So you need to really be careful and make sure your seeds are ripe. Otherwise, it can introduce more bitterness and astringency. So there are a lot of different techniques of extraction. So I want to just focus on one that is enzymes, because that's a tool you can add on grapes. So talking about the anatomy of berries, so that's a drawing of a berry. You see that the color and tannins are located in the skin. And you can also see that the cell walls of the skin of the cells in the skin are way thicker compared to the cell walls of the flesh. That means that you will need more action to extract these compounds. So the flesh is easily extractable compared to the skin, and this is where the interesting compounds are, the tannins and anthocyanins. So let me show you this trial. So this trial is a coloration trial. So it's a shift test colorant that shows the polysaccharides in the cell walls, so we can visualize them. So on the control, we can see that they are all in the color and the polysaccharides are all in the cell walls. So we need some enzymes to hydrolyze the cell wall to be able to extract as many compounds as we can. So with this specific enzyme, the enzyme crush, it breaks down the complex pectin matrix, but also the other pectin chains close to the skin. So it will help to lose the structure and release phenolic compounds, not only in the flesh, but also from the skin. We compared it with another enzyme, and we can clearly see that this enzyme X will only act on the flesh and leave much of the skin's cell wall intact. This is why it's important to really select a specific enzyme that will be able to extract compounds of the skin. So we see that the enzyme fresh both hydrolyzes the flesh and the skin layers. So using an enzyme will help to improve the phenolic and color extraction from the skin without being too aggressive in terms of tannins extraction or seeds extraction. And it's not only releasing phenolic compounds, but also aromatic precursors that we have in the skin, polysaccharides that are responsible for mouthfeel and the stabilization of wine in general. And it will also help to stabilize the color. So to show you another result, so that's from the same trial. It was on the Cabernet Sauvignon. This is liquid pectinase, an enzyme that you can just add on grapes to boost the extraction. So there is the result of the color intensity, but also the IPT. So IPT is the total polyphenol index, meaning that is the total amount of tannins extracted. So it's not only increased the color intensity, but also the overall tannins quantity. So using this kind of enzyme, with this one, it's very highly concentrated, so you can add a very low dosage, and so it's going to improve the color stability because it's, as I told you, it's also extracting polysaccharides. It will improve the mouthfeel and help to get more free-run juice. So it does extract more juice. And thanks to this enzyme, you will have less pectin and so less components that can impact the filtration and the settling. So a more clarified juice or wine toward the end of the fermentation. So then once we have our phenolic compounds extracted, the idea is to protect them because we don't want them to be oxidized or we don't want them to react with the proteins. So first of all, let's talk about the different oxidation phenomena, so you can understand what's happening and how to avoid the oxidation and the loss of your tannins and anthocyanins. So phenolic compounds on the grapes after being harvested, they can break down and okay, let me show you. So we have loss of phenolic compounds and color by enzymatic oxidation. So enzymes like lacase or PPO, polyphenol oxidase, can interact with oxygen and turn phenols into unstable molecule called quinones. It also, this kind of polyphenols can also react with oxygen and create quinones. So what happens to the quinones? They are very unstable and highly reactive. So first they can react with the glutathione, that is a molecule that is naturally found in the wine, so that will stop the oxidation chain, but they can also polymerize with other polyphenols and create browning, or with aromas. So when the polyphenols are reacting with oxygen or other enzyme, we are basically losing them because they can react with other polyphenols, or we can lose some other aromas. And then there is no recovery. That's forever. And then another way the phenolic compounds such as anthocyanins and tannins can be lost is by reaction with proteins. So the phenolic compounds react together, they can aggregate, and then they precipitate because there is an agglomeration between the proteins and the phenols, and they accumulate, and this is how you precipitate the color on juice even before the fermentation. So to avoid this kind of precipitation or oxidation, we have several tools. So you can use more glutathione, which is a natural antioxidant, SO2, but SO2 can actually bleach the color. So if you want to reduce the dose, but use alternatives. So using sacrificial tannins can be a good tool because basically they will protect the natural tannins of your wine, of your must, without impacting the must. So here is a little drawing to show you what's happening. So when we are adding sacrificial tannins, usually the pro-anthocyanidins are the one that are the most reactive with proteins. So they have a high affinity with proteins. That means that if you're adding these tannins on your must, they will react with the proteins, and it's not going to be your natural tannins that will be removed and won't react with the proteins. So you see that you're not going to have any remaining sacrificial tannins because they will be gone, because they will react with the proteins, but at least you will have a higher potential, phenolic potential, as I told you at the beginning, to be able to stabilize more color and have a better structure in the wine. So this kind of products, our product called Pro-Tannin R, it's very easy to use as you can directly sprinkle it on grapes. It will be directly soluble. You don't need to rehydrate it. It's also very useful if you have botrytis because it will inactivate the lacase and PPO activity. So on grape that is pretty healthy, you can use around 120 grams per ton. And if you have some damaged grapes, 180 grams would be a better dose. So as I said, it does remove the proteins, so it helps the clarification and to respect your natural tannins and preserve the grape potential. You can also reduce or replace the SO2 addition using it. So that can be a solution for you to just protect your phenolic potential at the beginning on juice or on grapes. Then I have another trial to show you. Same, it was a trial on Merlot to show you after malolactic fermentation how just using a sacrificial tannin will impact the color for longer term. So if you see the color intensity is higher as well as the quantity of tannins. It's clearly, we can clearly see it on the different colors of the glass. And the color stability is also improved, meaning that you're not only have better quantity of anthocyanins and tannins, but the color is also stabilized, which is also very important. So speaking of stabilization, we'll talk about the phenomenon of anthocyanins and color stabilization and any tools that we can use to actually keep the wine more intense until the bottling and until the consumers, the customers are opening a bottle. So in red wine, there are a lot of chemical reactions between anthocyanins and tannins. So color stabilization can occur by condensation. So it's the most stable color stabilization. So it's the stabilization between pigments and tannins. So I will go over the different phenomena, but the most stable and the reaction we want the most is this one. It's anthocyanin and ethanol and tannin complex. We call that ethanol bridge. So this will occur, this will work like it is. So the anthocyanins react with acetaldehyde, so ethanol, and the tannins to form a stable complex. So this will help to enhance the color stability and prevent any degradation. This is the most stable complex. So this is what we want to achieve. We also have anthocyanins and tannins complex with oxygen. Oxygen will promote the formation of stable anthocyanins and tannin complex. That will also provide a long-term stability. There is also a tannin anthocyanin complex without oxygen that can occur during the winemaking, and also color stabilization thanks to ellagic tannins, thanks to oak tannins. We can also have copigmentation. So copigmentation will boost color intensity and stability using hyperchromic or bathochromic effects. So that means that a pigment, so anthocyanins, and a copigment, which can be flavonols, manoproteins, or polysaccharides, will interact each other to create copigmentation. So the only thing is that these interactions can break apart over time, especially as the wine is aging. But copigmentation does enhance the color of young wines, giving a brighter and more vivid appearance. So that's all the different phenomena. And the idea is to make sure we can stabilize and create as much as this condensation or copigmentation we can on must during fermentation or even during aging.
Hey, Laura, you still have about 5 minutes.
Okay, I'm almost done. So this is a trial I want to show you. When you have a lower phenolic potential, you don't have, for example, enough color or tannins, then you can add some tannins during the fermentation to promote more of the condensation between anthocyanins and tannins. So adding more reactive tannin to the wine will help to stabilize the color during the fermentation and will help to have a better color. So soft tannin vinification is one of our tannin that is a blend between catechin, so reactive tannins, and polysaccharides to stabilize the color, get a better color intensity, and also impact on the mouthfeel. So basically on the first diagram, you see that over the time, 6 months after the fermentation, this Grenache lose way less color with the soft tannin vinification, and the overall tasting is better, more complexity, structure, softness, just adding tannins during the fermentation. So that's exactly the idea of tannin polymerization, to actually decrease the astringency and stabilize the color. So there are some tools like this that can replace a lack of tannins or phenolic potential, for example. So then I'm just going to talk about this one, as it's almost the end, to also be able to stabilize the color and build from some structure during the aging, it's not too late. There is still some possibility to use some grape tannins or some tannins to actually stabilize the color or help to get a better color by using this kind of tools. So usually I always suggest to add it when pressing or at the beginning of the aging, so the tannins can polymerize well in the structure of the wine and stabilize the color for long term. And using oak tannins will also help to prevent oxidation. So this is a combination of grape tannins and oak tannins. So grape tannins addition will help to create more stable color over the time, and oak tannin will actually prevent the oxidation of compounds. So really protecting the wines from oxidation, so from precipitating color, losing colors. And it does build for the rest of the aging, having a better structure. So you can use it with micro-oxidation, micro-oxygenation, for example, use at the end of the fermentation or during maturation regarding what you taste. I always suggest to do some bench trials for aging tannins because you don't really know how it will integrate with your wine. So from 5 to 20 grams per hectoliters, that's a good dosage. So finally, to sum up, from the grape potential to the crushing of the grapes, then using sacrificial tannins can help to use less SO2 or to just preserve the natural tannins of your wine. Then extraction is also a key moment to extract as many color and tannins, and then using of course enzymes, but you can also do more of tannins extraction, using maceration methods or any techniques that will work the best with your wines. Then for color stabilization, using polysaccharides that will stabilize the color, or adding more tannins to actually stabilize more anthocyanins will help. And then toward the aging, adding some oak tannins or using barrels and using more grape tannins will actually help to start with a better aging potential and not lose any colors during the aging. And then to not use SO2 or to reduce the dose of SO2 because it can definitely impact the color. Of course, you can't really remove the SO2 without finding an alternative. So using bioprotection for preventing the grapes against microbial issues, you can use bioprotection. Also, the type of yeast you're using during the fermentation will impact the production of more polysaccharides and color. So this is why it's important to think about which yeast you're going to use and what nutrition program. And also during aging, as it's usually the second addition of SO2 you're doing after malolactic fermentation. So to remove the SO2 addition or being able to lower the dose without having any microbial spoilage, using ketosane, such as our kilbrate, will definitely help to lower the dose of SO2 and have less bleaching during the aging, or at least will help you to postpone the SO2 addition during aging. So thank you very much. That was a lot of information, I think. But if you have any questions, please let us know. We are here to help. If you have any questions regarding tools or techniques during winemaking for any issues or enhancing a wine, let me know. We are based in California, but we have our products all over North America and Europe, of course.
Great. Thank you so much, Lora, for the great presentation. It was so little time to present everything, which is a challenge, but yeah, you did a good job just to explain us everything. So if you have any questions related to the presentation of Lora, please use the chat box to type your questions. We will answer your questions by the end of both presentations. So if you don't find the chat box, you should have 3 dots at the bottom of your screen, and you should be able to click on the 3 dots to use the chat box. So now it's my pleasure to introduce Dr. Catherine DADMUN. Catherine's background in both chemistry and French and Francophone studies from the College of Charleston in South Carolina was the perfect foray into her study of enology and wine chemistry. She completed her master's degree at Cornell University in New York in food science and technology in 2020 while working in Dr. Anna Catherine Mansfield's lab at the Cornell Craft Beverage Institute exploring anthocyanins in interspecific hybrid grapes. Following graduation, she stayed in Dr. Mansfield's lab to pursue her PhD, and she has recently successfully defended her dissertation and is graduating at the end of December. So now, congratulations, Catherine, to become Dr. DADMUN. Her PhD has been a collaboration between Cornell University in the US and the University of Burgundy in Franche-Comté in France under the framework of the French ISITE-BFC grant, an initiative for international exchange incorporating multidisciplinary approaches. During her 18-month research stay at UBFC, so in France, she worked with Dr. Régis Goujon and Dr. Natalia Quijada-Morin at the Institute of Vine and Wine within UMR PAM in Dijon. Her PhD research involved the exploration of interspecific hybrid grapes and wine, including projects on consumer perception of hybrid wine color, targeted and non-targeted metabolomics of the unique phenolic and anthocyanin profile of hybrids, and exploration of copigmentation in a hybrid wine matrix. Following graduation, Catherine plans to return to Dijon as a research engineer for continued work on color chemistry. Her greater aspirations are to address diversity and accessibility of science, as well as in the world of wine. Her passions lie in the interdisciplinary nexus of human interaction with food, wine, and science, including science communication and bridging the worlds of research and industry. Thank you very much, Catherine, for being willing to present your research project on color. So the floor is yours. You're on mute.
Sorry.
It was good. Yeah,
Sorry.
you were just on mute, but you were able to share your screen correctly.
Yeah, I couldn't figure out how to unmute myself while my screen was shared. I'm so used to Zoom and not WebEx, but this is fine. Okay, thank you. And thank you all for tuning in and your attention. And thank you, Lora, for an excellent talk and really great background information. And like Aude said, that is a really complex topic with a lot of different information. So you did a great job on getting all the topics included. So today I'll be talking a little bit about interspecific hybrid wines. And as Aude said, my research interest is largely in consumer perception. And so we'll talk a little bit about color management, but also about these projects we've done, trying to better understand color in hybrid wines, and then in turn, how to best manage them, how to optimize the color. As was introduced, my research largely was trying to bridge analytical chemistry and understanding the chemical composition of color and phenolic profiles and sensory experiences and sensory parameters of hybrid grapes and wine. And it's my understanding that many of the people here today might work with hybrids, so you're definitely better versed than me on making wine with the hybrids. So I look forward to the Q&A section and discussing that. But hopefully this will give you a little bit of insight into what's going on in the research world as well on color and hybrids. So just briefly, the topic I'll be covering today is about consumer perception of hybrid wine color, because we know that they have chemically some differences than Vitis vinifera does. And so we were trying to establish groundwork and explore if these unique anthocyanin profiles have a true effect on wine quality perception. So that's what I'll be digging into today, but just for your knowledge for the Q&A section and for future work, the other two topics that I worked on in my dissertation were characterizing recently released hybrid cultivars in Europe that were released in Switzerland and in France. And so because these were unstudied cultivars, trying to establish groundwork on understanding their profiles in order to eventually optimize color and phenolic treatments in the winery. And then Lora also mentioned co-pigmentation and color interactions and how critical they are in the context of wine color. So one of my projects was exploring co-pigmentation in these unique anthocyanin matrices that exist in hybrids. And I won't be discussing that today, but I hope one of the takeaways today will be understanding that the interactions that these anthocyanins go through, that these color compounds go through in the wine, is just as important as extracting the color itself. So like I said, I think many of you are probably familiar with hybrids. Just in case you're not, interspecific hybrids are called so because they're crosses of two or more species, Vitis species. Vitis is the genus, and Vitis vinifera is the traditional European wine grape species, what traditionally wines are made of. So these hybrids are traditionally bred crossing Vitis vinifera and other Vitis species in order to enhance the disease resistance and sometimes cold hardiness of the final hybrid cultivar. And usually that resistance is derived from the non-vinifera species. Sorry, I'm a little sick, so I hope that I won't be coughing too much. So like I said, it's been established that hybrid cultivars can have some compositional differences compared to Vitis vinifera, and therefore they can respond differently to traditional winemaking techniques. And then in response can also potentially affect the overall quality of the wine. So in the context of color, working with this research project that was in the Cornell Craft Beverage Institute, this project was based on the fact that there was anecdotal evidence that hybrid wines can portray unusual or unstable color, but what exactly was unusual about it was kind of undefined. And one of our observations was that it varies widely based on cultivar, as all winemaking does with Vitis vinifera as well. But usually the complaints were that sometimes the color would be much too intense for the other attributes of the wine. Sometimes hybrids could have a more blue hue than what is traditionally expected of red-purple in a red wine. And one of the big concerns is color loss over time, having a robust red color in a red wine, and then even over just a few weeks, losing that color, or having browning and things like that. And all of these things can also affect aging potential. So with all of these miscellaneous concerns and anecdotal evidence, we were looking at how to truly explore from like an empirical standpoint if these hybrid compositions do affect wine quality, and if so, what are responses we can do in order to optimize the color. So I promise I will leave as much chemistry out of this as possible, but because I'll be talking about anthocyanin profiles, I am going to explain some background, some base knowledge that might be review for many of you as well. So this 3-ring compound is an anthocyanin. I don't know if you could see my cursor. And so anthocyanins are, this is essentially an anthocyanin backbone. This is like kind of the base structure, and then it can have more things attached to it. And anthocyanins, of course, are responsible for color in red wine, and they are extracted from skins, as Laura mentioned as well, except for in tintoria varieties where they are accumulated in the pulp. And some hybrids, that at least the ones that I've worked with, some of them are tintoria varieties, which can lend to that really intense color. And as Laura said, again, color is a really critical sensory parameter because it's not only the first impression that a consumer gets of a wine, but studies have shown that visual perception can affect subsequent aroma and taste expectation and aroma and taste perception. And so all of these things together are not taken alone when a consumer experiences a wine. So this is why optimizing color in a product is really critical. So anthocyanins are inherently unstable molecules. So essentially they're prone to being attacked by other compounds where the other compounds can attach at one of these two sites. And if another compound interacts with it and connects to the anthocyanin, then this compound will be rendered colorless and no longer contribute color to the wine. And again, thank you for such an excellent, thorough talk, Laura. She mentioned the importance of color stabilization. And actually in a wine, these monomeric, just this single compound by itself, like 90% of the anthocyanins that exist in wine would be colorless in its native state. And so this is why connecting with other compounds, not in this way that renders it colorless, but in where it keeps it color and becomes stabilized, where we consider it stabilized when it's less prone to being attacked by these other compounds, then it can keep its color and remain in solution in a colored state. So there are 6 naturally occurring anthocyanin backbones found in grapes and wine, both in Vitis vinifera and in hybrids, all Vitis genuses, or sorry, Vitis species. And these anthocyanins range just in what is attached to this outstretched B ring here. So for pelargonidin, there's 1 OH attached, and then cyanidin has 2 OHs attached, etc. Delphinidin has 3 OHs attached. And this slight change in its structure is what changes the hue. And so these 6 anthocyanins range in hue from orange to violet. And although anthocyanin accumulation in the vineyard and extraction and stabilization during winemaking can be somewhat manipulated using different processing techniques, each grape cultivar generally has what we refer to as an anthocyanin fingerprint, so the types and proportions that that given cultivar accumulates. And in most wine grapes, particularly in Vitis vinifera, alvedin is the major anthocyanin that makes up this profile, which is why traditionally wines have a red-purple hue. However, some hybrid cultivars show major proportions of these other anthocyanins. For example, certain cultivars might have large proportions of cyanidin-based anthocyanins, and you can imagine that this might in turn affect the hue of the wine. The other major difference is that Vitis vinifera grapes accumulate almost exclusively monoglycoside anthocyanins, whereas hybrids can accumulate varying proportions of diglycoside anthocyanins. So the difference here is that monoglycosides have 1 sugar attached, whereas diglycosides have 2 sugars attached. And then the red R's are the ones that vary based on the backbone of anthocyanins. So to clarify, hybrids contain both mono and diglycosides, whereas Vitis vinifera only contains monoglycosides. And this simple compositional difference has the potential to affect color expression and color evolution, But exactly how it affects it and how that corresponds to consumer perception has been pretty unstudied. But this effect comes from the nature of diglucosides. So diglucosides are less pigmented than monoglucoside counterparts at wine pH. So that can be seen here, where Malvidin monoglucoside standard and Malvidin diglucoside standard are individually dissolved in model wine solution at the same concentration. And you can see the distinction of intensity of color and hue. Diglucosides are also more prone to being attacked and rendered colorless than their monoglucoside counterparts, and they form less polymeric pigment, which are when tannins and anthocyanins bind into a colored adduct that is more stabilized than anthocyanin alone. Laura also mentioned this interaction, and this is really critical in wines that are not young anymore, wines that are over a few months old. A lot of color forms into these complex anthocyanin interactions, and polymeric pigment contributes a kind of brick red hue to aged wines. But diglucosides don't form this polymeric pigment as readily as monoglucosides. So that's why aging potential in hybrid color can be a bit unique compared to Vitis vinifera wines. And then diglucosides are also more prone to browning than monoglucoside counterparts. And so you can imagine some hybrids have large proportions of diglucosides, some have only a little bit of diglucosides in their matrix, but this difference in composition has the potential to affect color expression and color interactions. Regarding the winemaking process, this was a study done in Italy on 2 different hybrid cultivars. The first Cabernet Carbon are the 3 top plots, and the second hybrid cultivar Prior is the 3 bottom plots. And the different plots there are just different years or different plots, and they were exploring extraction and loss rates during fermentative maceration of these anthocyanins. So as you're likely aware, anthocyanins reach a max extraction over fermentation, and then they begin to decline because anthocyanins are inherently unstable. And this, then when they start to interact with other compounds like tannins, then they start to stabilize. And what they consistently found across these many years and these 2 cultivars was that there was a 3-fold greater percentage decrease in the monoglucosides compared to the diglucosides. And so basically in the final wines, diglucosides were less prone to degradation following maximum extraction. And as I said, they also don't form stable adducts as readily. And so this can be of consideration if you are using a hybrid cultivar that has a high proportion of diglucosides. Following fermentation, you're likely to keep more of those diglucosides than the monoglucosides. A separate study done at within the Cornell Craft Beverage Institute, long before I was a member there, was on processing methods of 3 different hybrid cultivars, Marshall, Foch, Coro Noir, and Marquette. And the methods included pectolytic enzyme addition, exogenous tannin addition, cold soak, and hot press. And some of these methods were what Laura had also mentioned as traditional methods to optimize color. And what they found in these cultivars was that they successfully increased must anthocyanins, but they didn't see a corresponding increase or change at all in final wine anthocyanins. They also found, similarly to the last study, that 30% of the initially extracted monoglucoside anthocyanins remained in the bottled wine. So there was pretty significant loss of monoglucosides, whereas 80% of the diglucoside forms remained in the bottled wine compared to what was initially extracted. And I said hot press here. I think cold soak also showed a similar trend to this. And so in this example, the processing methods didn't have a significant impact on the phenolic profiles or color, but demonstrated again that diglucosides are less prone to loss compared to monoglucosides. All that being said, this example is not the final word that traditional processing methods won't improve color in hybrid wines. As Laura mentioned, it's so dependent on cultivar and temperature and method approach that it's worth trying some of these. But generally with hybrids, my understanding is that anthocyanin content actually is not really the concern. So many hybrids have really robust anthocyanin concentrations, but color stability is truly the challenge. And so when we think about color management and color optimization, it can't be dismissed that tannin management goes truly hand in hand with color management because of how critical those interactions are in maintaining color and preventing browning and things like that. And so many of you are likely aware, and this is if you work with hybrids, and unfortunately, we'll probably need another webinar to fully flesh out tannin management in hybrids because it's such a particular challenge. In many hybrid cultivars, they are prone to not retaining tannin. So many of the grapes themselves have tannin, but then they're not found in the final wine. And this is theorized to be due to binding with grape-derived proteins. And so generally it's suggested to do tannin additions after pressing off grape matter so that you don't lose proteins to binding with the, or sorry, you don't lose tannins that bind with the grape protein found in the grape cell wall, and potentially adding more tannin than, or more tannin supplement than is usually demanded of Vitis vinifera. But again, we won't dig too much into that, but it's just important to be familiar in how important these two management techniques are alongside each other. So as I said, there's still ongoing work to optimize viticultural and enological techniques using these disease-resistant cultivars because they carry these unique traits, and some of them don't respond normally to traditional winemaking techniques. There's a lot unstudied on how these unique compositions affect color parameters, color evolution, interactions and stabilization, or sensory perception. So in the case of my work, and what I'll be sharing today is trying to evaluate how this composition might affect consumer perception. So there was kind of anecdotal suggestion or assumption that these unique diglucosides that exist in hybrids but not in Vitis vinifera negatively impact wine color and wine quality perception. But there was no data on whether that was true or not. So this was one of the projects that we explored throughout my dissertation, was evaluating if the unique anthocyanin profile affects color expression, consumer perception, and overall quality perception. And before I dive into how we went about exploring that, I want to make a brief note about using Pinot Noir in these studies, because you'll see that we use a lot of hybrids and then Pinot Noir. And it's really important to note that Pinot Noir was not used as a standard or as a representative of all Vitis vinifera. In fact, we use Pinot Noir because it is unique among Vitis vinifera grapes in its anthocyanin profiles and accumulation. So specifically, it accumulates very low levels of anthocyanins compared to most Vitis vinifera red grapes. And it also does not contain acylated anthocyanins, which are just an anthocyanin derivative that are slightly more stable and make up significant proportions of most Vitis vinifera anthocyanin profiles. And furthermore, this project was a collaboration between Cornell University and the University of Burgundy. And Pinot Noir is at the heart of Burgundy's wine industry. It makes up almost 90% of the red grapes grown in Burgundy are Pinot Noir. And so in my experience, it's challenging to even do any projects in Burgundy without using Pinot Noir. But keeping those caveats in mind, please don't look at it as we were comparing Pinot Noir as the ideal Vitis vinifera or anything like that. So, and during the Q&A section, we can discuss more about that as well. So for this work, 17 commercial wines were sourced, including 14 hybrid varietal wines from all over the United States and Pinot Noir from 3 representative regions: Oregon, the Finger Lakes in New York, and Burgundy in France. All of these wines underwent instrumental analysis, and a subset of 8 wines that are in the top table here were part of the sensory study. And because hybrid cultivars are very diverse depending on their parentages, we aim to get a variety of types of hybrids. So we have Vitis riparia-based cold hardy under this section of category, French American, Neo-American, and German American hybrids. So this sensory study took place at Cornell University Sensory Center and included 82 pre-screened general consumer panelists. They were first given a wine with a cover over it and asked to rank on a hedonic 9-point scale how much they liked the overall appearance, the hue, and the color intensity of the wine, and then how much they expected to like the wine based on visual attributes alone. Then they were instructed to remove the cover and smell and taste as they normally would, and rank how much they actually liked the wine. Finally, we also asked consumers to rank how well the aromas and flavors matched visual expectations, because we hypothesized that if there was a schism between the smell and taste of the wine compared to the color-based expectation, then the overall impression of wine quality could be affected. Excuse me. I'm going to get a sip of water.
You're going to have 5 minutes remaining, Catherine.
Okay. So the instrumental analysis included C-Lab colorimetry, which, if you're not familiar with, is a method of analysis using 3 dimensions to try to replicate human perception of color. So the first parameter is L star, or lightness, that goes from white to black, A star, which is green to red opposing values, and B star, which is blue to yellow opposing values. And we usually visualize the C-Lab color space like this, but truly it more looks like this on the right. So it's a bit of a complex color space, but it's really widely used in the food and wine industry. And then using these values, we can calculate hue, which is the tint or shade of the wine, and chroma, or color intensity, which is the saturation of color. We also measured absorbances at 420, 520, and 620 nanometers using UV-vis spectrophotometry, total anthocyanin concentration, and we also isolated an anthocyanin extract from each of the wines, which was analyzed on ultra-high pressure liquid chromatography. And basically all these different peaks were individual anthocyanins that we can then separate and quantify and make up the entire profile. So the first thing we found from the sensory study was that these 3 visual parameters, could all be collapsed under overall appearance liking. So this just demonstrated that consumers used color in a holistic fashion rather than consciously separating factors like hue and color intensity. So this graph here shows average results for the 8 sensory wines of overall appearance liking, expected liking, and actual liking. And we found that overall appearance liking showed a significant relationship with expected liking. So this implies that the higher consumer ranks visual characteristics, the more they expect to like the wine. Notably, there was a separation of a higher ranked cluster for overall appearance liking and expected liking, and a lower ranked cluster for those 2 purple and green values. And it's important to note that actual liking, the pink bars, did not follow this separation of clusters, and Pinot Noir is dispersed between these 2 clusters. And so the actual liking could not be predicted by either overall appearance liking or expected liking. And this made a lot of sense because factors beyond visual perception, like taste and aroma and mouthfeel, carry much more weight in a consumer's final quality perception analysis of a wine. And then we found some interesting connection between expectation matching, the rating of how well aroma and flavors matched expectation based on the wine's color. This showed a significant relationship with the difference between expected liking and actual liking. And so this suggested that if aromas and flavors don't match color-based expectation, then this could be a predictor for how much final quality analysis changed from initial visual analysis of the wine. So this is a chart that shows the 3 dimensions of C-Lab collapsed into 2 dimensions. So usually it has L, A, and V. And here we have L star as the y-axis, lightness, and then hue angle, which is calculated using A and B on the x-axis. And pretty much the takeaway here is that those 3 less liked wines for their overall appearance and expected liking were kind of separate, with including 2 outliers compared to the other sensory wines that were higher ranked. And we see a wide diversity in color parameters of all of the hybrid wines, including those that weren't part of the sensory study. But those largely fall closer to those higher ranked wines than the lower ranked ones that had high lightness and high hue value. And then when we look at the colors of the wines ranked from least liked to most liked, we could see that the lower ranked cluster above the yellow line shows lighter colors and more brown hues compared to the purple hued, more intense colors of the higher ranked wines. And this was corroborated by the fact that lightness or luminosity L star, hue calculated by both colorimetry and spectrophotometry, and yellow tones, which is measured using B star, all predicted overall appearance liking and expected liking. But none of these visual attributes corresponded at all with actual liking. And this is consistent with other sensory evaluations of color of Vitis vinifera in the literature, where generally darker colors are more liked, and less brown tones are more liked. And so finally, this is just a distribution of the anthocyanin profiles, and we'll just look at the red and green bars. So red is monoglucoside proportion, and green is the diglucoside proportion. And what you could see is that, of course, the 3 Pinot Noirs here don't have any diglucosides in them, as expected. But the other hybrids show a wide range in diglucoside proportion. Some have a wide majority of diglucosides and some don't. But importantly, we see that for the 2 wines that were the highest ranked for overall appearance liking and expected liking, have extremely different anthocyanin profiles. So the 2 highest ranked were St. Croix and a hybrid, and Pinot Noir from France. And St. Croix has a very high proportion of diglucoside content. But both of these wines were highly ranked and present dark purple hues that were liked by consumers. So this suggests that diglucoside ratio does not necessarily have a negative impact on wine color quality, and that diglucosides are not necessarily a bad part of hybrid color composition. So just to briefly summarize, color is an important predictor for how much a consumer expects to like the wine, reiterating how important color management is, but how much they actually like the wine depends on many other factors. Excuse me, matching of, oh, sorry about my cursor. Matching of aromas and flavors to color-based expectation has a really interesting impact in the change of perceived quality expectation to final quality judgment. So generally, we can now conclude, based on this data, this suggests that wines with diglucoside proportions can still exhibit desirable wine color. And this, again, mimicked work done on Vitis vinifera, where dark color and low yellow-brown tones are more liked. Finally, as I mentioned, cultivar largely determines hue and color intensity in the wine, but environmental parameters and processing techniques can have a large impact as well. Luckily, hybrid cultivars lend flexibility to wine style experimentation because they don't necessarily have consumer expectation that comes with some vinifera cultivars. So it can be beneficial to develop wine styles based on cultivars' inherent attributes rather than necessarily trying to fit it into a box. And this is kind of in the same vein as matching color to taste and aroma expectation for a consumer. So if it is not a very robust tannic wine, then a consumer might expect a lighter color. So keeping those things in mind during color management can be important and interesting. Although research shows that consumers rank darker colored wines more highly, it's really important to compare if wine color matches aromas and flavors. Sorry, the last thing is that considering cofactors, meaning the compounds that interact with anthocyanins, is just as critical as the anthocyanins and their extraction themselves, especially in the context of a hybrid cultivar. So thank you so much for your time, and I look forward to any questions and discussion we might have.
Awesome. Thank you, Catherine. It was a great presentation. As a reminder to all of our participants, the folks watching, Please type any questions you have in the chat box. Again, just click on the three dots at the bottom of your screen, and we will have our panel, and Ode and I will do our best to answer those questions. We're now going to be joined by Anne Zwink from Soldier Creek Winery in Iowa, Mark Germany from Fireside Winery, also in Iowa, and from my old friend Bryan Forbes from Gusmer Enterprises to discuss any Q&A or discuss any information that was presented. Ode, do we have any questions in the chat box?
Yeah. Thank you very much, first, Catherine, for all those explanations. And that's a topic, as you said, that could be 2 webinars, and we can see that with the time. But Catherine, there is one question for you. Have you considered doing PCA analysis between consumer results and quantitative data to find or determine a robust metric that drives consumers?
Yeah, great question. PCA analysis was not part of this data analysis. And I think we have discussed already what the next project will be in order to dig deeper into these questions of consumer perception and hybrid matrices. And like I said, this was kind of an assumption that was not studied at all. There's no data on it. And so it was almost an exploratory study, this one. And so I think that would be really interesting and critical, is to do a multivariate analysis to be able to kind of pinpoint what might drive consumers. I agree. So hopefully in the future we will do that.
Thank you. Maybe we can turn the microphone to Mark, Anne, and Bryan. You all work with wines that are hybrids, but also vinifera. Can you share your experience towards color management that you have in your winery? Any challenges?
Yeah. So I was, I was a winemaker in Texas before I moved here to Iowa, and tannins were an issue for us in Texas, not necessarily color, but getting stable color was more of an issue. So fortunately, when I came here, I was a little bit more in tune with trying to get stable color. One of the first things that I put into practice when I came here was cryomaceration, which we didn't really discuss in there. And it takes cold soak to the extra level and actually freezing the grapes or the grape must. Unfortunately, is a very risky process, but the great thing about it is you're going to freeze those cell walls, and by doing so, you're going to break open and extract pretty much everything. It's more effective, we found, than just pectic enzymes. The other thing that I found with these hybrids and moving here and working with these hybrids was almost like becoming a winemaker all over again. It was quite the experience, and still is. We're still constantly learning. But tannin products have become pretty much a way of life in this building for our dry reds. Our sweet reds, we're not going to put as much effort into it because we've found that our customers really aren't motivated as much by the color as a dry wine or a dry red drinker would be. But for our dry reds, max dose of sacrificial tannins, as well as cellaring tannins, we have found are extremely important in maintaining that color for a long period of time in barrel. And then on select wines, we don't do this on all of our wines, but finishing tannins we have found are extremely beneficial for wines that we know are going to be in bottle for a long time. I know that may be taboo to some winemakers to talk about, using these products, but they're a tool in the chest. Just like, I'll even mention it. We don't use this product here, but Mega Purple is a product that you could use. But I do encourage people that are going to use that product to use it very smart. The best place to use it's going to be pre-fermentation. If you do have to make a post-fermentation add, you have to make a tannin addition in order to keep those colors stable for a long period of time.
Yeah, thank you so much.
Good advice.
Especially for the cryomaceration. So that's something we work together on, and looking at just the results we had. So we did use Market and we did use Frontenac The color was extracted at the beginning, but at some point, same as tannin. So everything is getting extracted because you break all those cells. But at the same time, if you don't add other tannins, that change is not going to last over time. And so that's where doing a combination of different tools, that's how you can improve the color stability. Exactly what Lora, you explained about extraction. That's good to extract, but you need to protect and you need to stabilize all those compounds.
You know, if we could afford the equipment, flash détente would be the way to go. But you know, cryomaceration, we've got a freezer out back, and it does the trick.
Yeah. And do you have anything to share?
We use enzymes. We use Color Pro for during fermentation to help stabilize and extract, and we also do use fermentation tannins as well. And like Mark, we also in some of our select dry reds, we will add additional tannin before the bottle. I'm really glad, Catherine, that you spoke a little bit about Teinturier varieties, because all of our reds that we have here, if anything, we're usually talking about rosés and how to remove color a little bit. That's how I feel anyway. But of course, yes, it's important to keep the color in the reds that we have.
We have a question quickly on the chat. So Lora, you mentioned 120 grams per ton of sacrificial tannin to use. And so Kevin have found hybrids need a much higher dosage. He uses 400 grams per ton. So what are the others' experiences? Are you using much more than what Lora you mentioned?
I will warn you that there is a diminishing return. You know, there is, you can throw tannins at these hybrid varieties all day long. And it's almost, you know, set up for you to want to do that. But at some point, you've got to figure out where that matrix is, where you're actually doing benefit to the wine and when you're just throwing money down the drain. We have found at about 200 to 250 grams per ton is kind of our sweet spot. We've gone as high as 400 grams per ton before, and we really saw no difference. So it just depends on, you know, if you really think you're, I mean, data's the end all be all of all of this. So, you know, unfortunately, we're not able to, you know, collect all that phenolic compound data on this stuff. But just through analysis and the overall end product, we're able to really try to figure out what that diminishing return is. And we, like I said, we've found that around 200 to 250 grams per ton. And it also depends on the specific product that you're using. But at the end of the day, it's just, I mean, if you want to throw money at it, throw money at it. But you've got to really figure out that balance for it, for sure.
Bryan, I'm glad you're back. What has been your experience here? Most of your experience here in the Midwest.
Let's see if you can you hear me at all?
Yes.
Yeah, you can. Okay, cool. Sorry about that. I'm on a ferry in the middle of Long Island Sound right now. So.
Oh.
We lost you, Bryan.
That's too bad.
Okay.
Bryan and Mark and myself all have experience, and well, all of us have experience with both vinifera and with hybrids. I was looking forward to Bryan's input or comments. Lora, is there any plan for you folks to do any experimental winemaking with hybrids? I assume all of your data was mostly from vinifera.
Yeah, absolutely. That would be, so we have an experimental center in France that is developed for this kind of trials. but we also do a lot of on-field trials in the US, so that would be definitely a subject for the next harvest for sure.
Oh, good.
Yeah.
I do have a quick question, maybe for you, Catherine. That's when you said that the ratio of diglucoside versus monoglucoside doesn't really impact the color liking of people, but you still observed that Saint Croix, which is really rich in diglucoside, and Pinot Noir, no diglucoside, are the most liked wines. Did you look at the type of forms of anthocyanins that you had? If it was more of one structure compared to another structure? And also, did you compare, or did you look at the vintage of the wines?
Yeah, good question. Oh my gosh, I'm so sorry. Yeah, so with that anthocyanin extract, we were clarifying what forms of anthocyanins were part of that profile. And in all of the cultivars that we explored, none of them had large major proportions of non-malvidin anthocyanins. So malvidin, mono, and diglucoside still made up the majority of the proportion for all of these cultivars that were part of this study. And then regarding your question on vintage, this particular study was only done based on one vintage of wine. And as we all are probably very aware, vintage has a huge impact on every parameter you could possibly measure. And so it would be really interesting and relevant to explore further. And the other thing is that we sourced commercial wines because we were hoping to kind of replicate what a consumer would find like in the wild. But because of that, we couldn't control winemaking parameters, and we also were sourcing them from different parts of the United States that each given environmental environment would affect how that cultivar is expressed, how it accumulates anthocyanins, et cetera, et cetera. So those factors that weren't controlled also are parameters that this kind of research should be continued in order to clarify. It's maybe not all St. Croix have that kind of hue and that kind of color that people like, but part of the challenge for what we were challenging in this study had a lot to do with OIV regulations, so the International Organization of Vine and Wine, that of course the United States, except for Texas, if I understand correctly, is not part of the OIV. So I didn't think it was relevant to talk about today, but especially with the work in Europe and that they're releasing hybrids to be used in the wine industry, but the OIV still does not allow diglucoside content over 15 milligrams per liter, which is a very low threshold. And there's even some misinformation being spread about like a health impact of malvidin diglucoside. So really bizarre stuff that I think we were trying to get empirical data to say, actually, they might not be contributing to a negative wine color. But that being said, as I showed, they do tend to brown faster or have different interactions with other compounds in the wine matrix. And so it's still worth the consideration of these different composition, but it challenges that idea of they're inherently bad and inherently worse anthocyanins or something like that. So I hope that answered your question. I don't know.
Yeah, it does. And I think that would be very interesting just to look at the same wines, but 2 years later. How is the color gonna change, and especially how consumers are gonna like those wines? Are they gonna like the same? Are they gonna rate those wines in the color in the same way, or is that gonna change?
Yeah, I don't think I included it in the methods, but these were wines that were within like 1 or 2 vintages, so they were relatively young wines.
What vintage was it?
Specifically. Sorry?
Which vintage was it?
It was 2019, so we were sourcing wines from 2019 or 2020 release, and ideally not beyond that, because we were trying to at least control a little bit that vintage question of how long has the wine been sitting in bottle or aging, things like that. Yeah.
You know, something I've had to do as a winemaker here in the Midwest is just come to terms with the fact that not every one of my wines is going to look like a Petit Syrah. You know, it's just not. But and it's very frustrating. Frontenac is probably the most frustrating grape that I work with color-wise, and it wants to brown very quick. I feel like we have Marquette pretty much mastered, but Frontenac is the one that is constantly given us the struggle. You know, I think, and you know, something that I did back in Texas a lot, I saignéed and bled everything. I did a 20 to 25% bleed on every red variety that I produced. I'd get a really great rosé out of it in the long run. Here with us, we're going to get darker rosés. That's what PVPP and activated carbon are for. You know, do with that as you please. But I do truly believe that even as low as a 15% bleed or saigné on your reds is going to increase that juice to skin contact ratio, and in theory should give you quite a bit more stability. With us, we're 100% mechanically harvested. Fortunately, our vineyard is only, you know, a 10-minute drive away with a full load of fruit. And, you know, hopefully one day I'll get our, you know, our head winemaker on board with doing a saigné. But I do think it's something for some of the smaller operations out there that if you have the ability to do that bleed, really look into it, because I had great success with it, especially with grape varieties like Mourvedre and Grenache that are really difficult to achieve the color that you're and the ripeness that you're looking for. Had great results with saigné on those.
Yeah.
Oh yes, I did a lot of saigné with both of those varieties in California. That's for sure. Well, Ode,
Yeah.
we're at 4:31.
Yeah, we have to conclude. Unfortunately, we don't have that much time to continue the discussion, but hopefully it's going to be a next webinar next year. So thank you very much to both presenters. Thank you, Lora, so much. Thank you very much, Catherine, for both of your great presentations. And thank you, Mark, Anne, and Bryan. So we didn't hear you, but stay tuned for next year webinar series. If you want to have this webinar series continuing, please suggest any topics. That would be very helpful. So send us an email and complete the survey that you should receive the link in few minutes. And so also I would like to thank you again, and I hope we brought some new knowledge to you today about red wine color management. So stay tuned for next year. Feel free to watch the recordings of previous webinars that are available on YouTube. And happy holidays. Have a great season. And
Hey,
great
thank
vacation.
you, everybody.
Thank you, everyone.
Thank you very much.
Thank you,
Thanks.
everyone. Happy holidays.